Hi, everyone. So this is Group 3's PowerPoint presentation on the role of acetyltransferases for the temporal-specific accessibility of beta-catenin to the myogenic gene locus. And presenting is Adriana, Alexis, which is myself, Dyanna, and Malena. Here's our agenda. So for the background, the role of beta-catenin is to express genes in the promoter regions, but they wanted to know how this was accomplished in the myogenic proliferation and differentiation stages by beta-catenin. So in the study, they showed how accessible beta-catenin was to the promoter regions of the target gene with the regulation of stage-specific HATs, which are histone acetyltransferases. And these are the lysine transferase 2B, which is CAT2B, and camp response element binding protein, CREB, binding protein, or CBP. In the WNT pathway flow, the beta-catenin is the key effector responsible for the transduction of the signal to the nucleus, and it triggers transcription of WNT-specific genes responsible for the control of cell fate decisions in many cells and tissues. So for the genes, it's CCNA2, which is cyclin A2, which is expressed during the mitotic division of somatic cells, CDC25C, which is cell division cycle 25C, which is a protein coding gene that is a regulator in the cell cycle progression, and then FERMT2, which is fermentin family homolog 2, which enables actin filament binding activity. So the role of the pathway is the WNT beta-catenin signaling promotes myoblast proliferation and differentiation by regulating these genes necessary to carry out these functions. So the phenotype or the molecular mechanism, the pathway is triggered during either the proliferation or differentiation stage of the myogenic pathway. They found that the HATs, CAT2B, and CBP were co-activated with beta-catenin during their assigned stages on the specific genes. CAT2B and beta-catenin were on CCNA2 and CDC25C during proliferation, and CBP and beta-catenin during FERMT2 differentiation. So it is possible for other epigenetic factors such as pollutants and toxins to find their way into our cells. They could potentially disrupt our WNT beta-catenin signaling, causing the expression of myogenic proliferation differentiation, or as we have learned in other papers, the deregulation of the WNT and beta-catenin signaling pathway can potentially lead to cancer and non-cancer diseases. So the issue to be solved is they wanted to know how beta-catenin was activating their target genes during the myogenic stage and proliferation differentiation stages, and if acetyltransferases were involved, and if so, it was continuously or just specific times. So was it just during proliferation and not differentiation, or vice versa, or was it continuous out the whole thing?
The scientists' collective goal in the study was to investigate CAT2B and CBP at different stages of muscle proliferation and differentiation. They wanted to see how HATs would be able to control gene expression during myogenesis and their chromatin accessibility. To do this, they used many different methods like QRT-PCR, HIP-qPCR, and immunoblotting analysis, which is Western blotting. They used these methods to find the binding patterns of certain gene promoters, and they also analyzed the relationship between HATs and beta-catenin. They then used the information that they got to get a better understanding of muscle development and healing, which is what they wanted to accomplish. The flowchart on the right shows us the steps and what they wanted to achieve. So let's go over it. They saw that beta-catenin was activating the target gene during proliferation and differentiation. They found this by seeing if Wnt beta-catenin signaling during the stages of proliferation and differentiation would bind to the target gene promoter regions, and if it would affect the gene expression. They then saw that during the different stages of cell proliferation and differentiation, what histone acetyltransferases were expressed. After, they were seeing the relationship between beta-catenin and HATs, and also the effects of IWRI endotreatment on gene expression during proliferation and differentiation. They then finally saw the effects of gene knockdowns on gene expression and the different levels of proliferation and differentiation in C2, C12 cells. What they came up with was that CAT2B and CBP play a role in regulating Wnt beta-catenin in muscle development. To do some experiments, we will need to have some background knowledge, more specifically for hypothesis-driven experiments. We will have to have some knowledge of the Wnt beta-catenin pathway You should know that the canonical Wnt pathway, also known as the Wnt-B-catenin pathway, involves the nuclear translocation of B-catenin and activation of target genes via TCS and lead transcription factors. You would also have to know temporal specific expression patterns of CAT2B and CBP during differentiation. CAT2B and CBP expressions vary at different stages of development as they are active and influence the expression. This information is important to know to be able to successfully understand the experiment.
The most important establishment experiment in our paper was figure 4A. Researchers wanted to determine the significance of CAT2B and CBP in the regulation of CCNE2, CDC25C, and FERT2. The question was if knockdown of CAT2B gene would result in the decrease in expression for genes CCNE2 and CDC25C in the cell proliferation stage. The method that they used was quantitative RT-PCR, and this was performed to measure cell proliferation stage. The total RNAs are isolated from C2, C12 cells and were treated with IWRI endo or the control vector for 24 hours, CAT2B siRNA or control siRNA for 24 hours. For the result, they noticed that when CAT2B siRNA is present, there is a decrease in the amount of CCNA2 and CDC25C DNA present in the sample. CAT2B knockdown siRNA also decreased the amount of CAT2B DNA in the sample. For the control was siRNA control, and that was used because it should not have affinity for CAT2B mRNA sequence, and it should decrease CAT2B protein levels. It was a control for specificity. So the authors were able to make a few conclusions. One being, since the addition of the inhibitor CAT2B led to a decrease in the amount of CDC25C DNA and CCNA2 DNA produced by the PCR machine, the researchers could conclude that CAT2B expression is required for the expression of CDC25C and CCNA2. Having less DNA product at a certain time meant there was a smaller initial amount of mRNA present in the sample. It was evident that CAT2B inhibition resulted in smaller levels of CDC25C and CCNA2 expression. For figure 4A, it was classified as expression and establishing. Figure 4A was considered an establishing experiment because they established that there is a relationship between CAT2B knockdown and the regulation of CCNA2, CDC25C, and FERT2. This information led on to experiments later on. For example, researchers used this information from figure 4A to test the role of CAT2B on the binding of beta-catenin, the promoter region of CCNA2 and CDC25C in figure 4E. Our group decided that figure 3E was the most hypothesis-driven experiment out of the paper. The researchers of the study were already aware that expression of genes CCNA2, CDC25C, and FERT2 were reliant on the Wnt/beta-catenin pathway. However, researchers were interested in how beta-catenin is able to target and promote expression of certain genes in a stage-specific manner. The scientific question: researchers wanted to determine the specific binding interactions between HATs, such as CAT2B and CBP, and beta-catenin during different stages within the cell cycle. The approach used was an IP immunoblotting analysis. The result: researchers observed specific protein-protein interaction between CAT2B and beta-catenin during the proliferation stage. They observed interaction between CBP and beta-catenin for cells in the differentiation stage. The controls: the IgG antibody was used as a specificity control. So what are the main takeaways in conclusion? They were able to conclude that beta-catenin forms a complex with different HATs in order to activate transcription of genes that promote proliferation, CCNA2 and CDC25C, or differentiation, FERT2. This is considered a hypothesis-driven experiment because they hypothesized a molecular mechanism on how beta-catenin interacts with CAT2B and CBP and proved that their hypothesis with the experiment in figure 3E. As stated by the authors, taken together, our results indicate that beta-catenin forms a complex with state-specific HATs in the nuclei, so that Wnt/beta-catenin pathway signaling can control gene expression on demand.
Our validation experiment is known as a pull-down assay. This is an appropriate method since it assesses protein-protein interaction similar to the IP immunoblotting analysis. In this case, a protein of interest, such as CAT2B, is immobilized and bound to an agarose bead. This bead is then treated with a cell lysate containing all proteins from the cell, including beta-catenin. It is then washed. This removes all proteins that did not bind. If there's indeed protein interaction between CAT2B and beta-catenin, it can be visualized using an STS page, which separates the proteins by size. In order to confirm the presence of proteins within the sample, a marker/ladder can be used to match protein sizes for identification. Since the collected product depicts two bands that align with CAT2B and beta-catenin, they can conclude that they were present in the sample and did indeed have protein-protein binding interactions within the proliferative cells. Our first experiment includes testing binding affinity between CAT2B and beta-catenin. Our second experiment includes testing binding affinity between CBP and beta-catenin. Within both of the experiments, it is observed that the final product of the pull-down assay contains both proteins, indicating a protein-protein interaction between the HATs and beta-catenin. Thank you for listening to our presentation. Here are our citations.